So today we have the pleasure of having two very fantastic speakers. I will introduce the first speaker, Professor Erik Santiso, who is an associate professor of chemical and biomolecular engineering from North Carolina State University at Raleigh, I think that's how it's pronounced. So Erik obtained his bachelor's degree from Universidad Simón Bolívar in Caracas, Venezuela. And he was, if I remember correctly, you taught at the university there. Yes. But things went south there, so he had to come to the US. He did his PhD at NC State with Keith Gobbins. Then he moved to MIT and he did a postdoc on studying different aspects of crystallization. And he basically started his own independent career at NC State in 2013. And he does a lot of work on studying crystal nucleation, particularly in fairly complicated systems like pharmaceuticals and organic molecules that have, very complex phase diagrams and nucleation pathways. And I have been strongly impacted by the work that Erik does, which is always superb, and we are really delighted to have him today. So without further ado, I will stop and I will leave the floor to Erik to talk to us about the role of solvent in crystal nucleation from solutions.
Okay, let me share the screen. Can you see it?
Yes.
All right. Well, thank you so much for the invitation, and thank you, Amir, for your very kind words. I hope I will live up to the expectations. I have to say this talk is going to be a little strange because, half of it is a story that is more or less reasonably complete, and the other is a mess because it's something we started more recently, and, I'm not sure I even understand everything. So, if I leave you with more questions than answers, that's probably because I also have more questions than answers. But let me start. So I want to talk a little bit about crystal nucleation, and you've had already people here who talked about this, so I probably don't need to say that much about the importance. But the motivation for us is looking at nucleation of small organic molecules, and those are of relevance to a number of different applications, including things like food, specialty chemicals, and in particular pharmaceuticals. And I would say probably 2/3 or so of the funding for the sort of work we do has come from pharmaceutical companies because, as you know, this is an important problem for them. So what I want to do here is, we have three major challenges that we're thinking of addressing here. And then one of them is the idea of polymorph prediction, so predicting crystalline forms. We had Claire last time here, so you probably heard about this problem. I am going to assume in what follows that problem has been solved. Okay? So there's a lot of really smart people who've been doing this for a very long time, and they have figured out ways to do it. So I'm going to assume that we're given some molecule, some drug-like molecule, and we have run that through some really amazing crystal structure prediction software, and we know the solid phase diagram. And now we want to answer questions such as, how easy is it going to be to crystallize this molecule? And under which conditions am I going to get one polymorph or the other polymorph, and things like that. And this is useful, particularly if you're in the situation where, let's say that you have some drug candidates that came out of a simulation, some docking simulation or whatever, and you're trying to decide which one is going to be easier to turn into a drug. If we're able to make these sorts of predictions, then we can make decisions as to, which one are we going to target for development first, right? Instead of having to synthesize everything and then run experiments. So we're going to be talking about these two problems, and the methods that we use to do this, is the string method in collective variables, which you may have heard of. And the idea here is that we have some free energy surface, and we're trying to find what is the minimum free energy pathway connecting to stable basins. So this is kind of a cartoon that is taken from Erik Santiso's website. And the idea here is we have two minima, and we're starting with a series of replicas of the system that are at different points along this free energy landscape. And we're going to try to evolve them such that they lie in approximate way on the minimum free energy path. Sorry, I keep hitting the wrong button. So the idea here is we start with this discretized path, and at every point on the path, we run a restraint dynamic simulation, and by computing the average restraint force, we get an estimate of the gradient of the free energy. And now we know which way the free energy goes down for every image, so we take a little step in that direction. Now, if we just iterated the process in this manner, what's going to happen is that the images are just going to collapse on the stable basins, and I'm going to learn nothing about the mechanism, right? So instead of just keep doing that, we take little steps, and then we interpolate a line through the new images, and we redistribute them along that line in order to keep some of them in the middle. So there are multiple ways you can do that. I mean, the one naive way is to keep them at constant distance from each other. What we do is once we have something that is reasonably converged, we try to concentrate images on the nucleation branch because that's the part that we're interested in. But you basically need to do that. And as you repeat that process, eventually that string converges to something that is, again, wrong button, an approximation to the minimum free energy pathway. And then we can start asking ourselves questions about, what is the mechanism by which this is happening? What is the free energy barrier, and so on. And in particular, the free energy barrier is what we use as a sort of metric to decide how easy it is to crystallize a molecule, and between if you have different conditions and which one is it going to crystallize more easily, and so on. So this is the method we use. And now, of course, in order to draw this free energy surface, you need to say what the axes are. And, typically those could be order parameters, something that tells you whether your system is crystalline or not. However, for organic molecules, it's not obvious what is a good way to construct these order parameters because, they are complicated and they make different crystalline forms. And what we've done is basically come up with a general way to construct this. And, I always show this picture because it's kind of fascinating, the fact that, the human brain instantly recognizes symmetry without you doing any calculation. So if you look at these two pictures, your brain in less than a second knew which one is a crystal. But when we have to explain it to a computer, what we have is the list of XYZ coordinates of the atoms, right? And then we need to construct some function that tells us which one is the crystal. And in general, it's not trivial. So the way we do it is by constructing a model for this kind of generalized correlation function, which includes information about, given a molecule, how far the neighbors are, but also in which direction, and how are those neighbors oriented with respect to the central molecule. So what we're doing is basically replacing this complicated molecule by a point, which can be the center of mass, or it can be a reference atom in the molecule. And we look at the vector joining that center to the centers of the other molecules. And we also use a quaternion to describe how you need to rotate one molecule to get the other. So every molecule has a little internal coordinate frame. And we just basically ask ourselves, how do I rotate this coordinate frame to get to the other? So this is something we can estimate from a molecular dynamics simulation. And in principle, if your molecule has internal degrees of freedom that can change, you can also incorporate this. I'm not gonna talk about that in this talk, but it's something that can potentially be incorporated if needed. Of course, if your molecule becomes ridiculously large and you have a lot of those, then this is perhaps not the best way to do it, because you would be saying that your very large, complicated molecule is a point. But for small, medium-sized organic molecules, once we've looked at it, seems to work pretty well. So we're going to construct a model for this, and the way we start is we say, well, we know what that function is at 0 Kelvin, because at 0 Kelvin, everything is frozen in its equilibrium position. So it's just a sum of delta functions. So what we do is we say, well, if the temperature is not too high, I'm going to assume that I can write this as a sum of products of functions. This sum runs over the peaks in the original function. So if you were thinking of just a regular GeoFR, you'd basically be adding Gaussians at every point where you have a peak in the GeoFR of the solid. It's just this one also includes orientational information. And then we construct models for these functions by looking at the crystal and seeing what they look like. So we postulate some forms for this model. So for the distance we use Gaussians, for the orientations and quaternions we use distributions that are suitable for those degrees of freedom. And we obtain the parameters that appear in that in those distributions by running an MD simulation and doing the usual maximum likelihood estimation. One problem, and this is a note that I'm making at the bottom because we're going to come back to this, is that this requires assigning the data observed to individual peaks. So when I have this index alpha here, I'm saying, I had this, what I call peak number 1, peak number 2, peak number 3, and so on. Let's say those are the ones at 0 Kelvin. When I am running the simulation, I have molecules moving and fluctuating and so on. And at every point that I'm collecting statistics, I need to figure out which neighbor to assign to which peak. And you cannot just simply say, well, I'm going to take the nearest neighbor, next nearest neighbor, and so on, because the molecules are fluctuating. And then what is the nearest neighbor now may not be the nearest neighbor a few time steps down the way. So the way we figured out how to collect statistics on the correct bin for solids is we first minimize the crystal, at 0 Kelvin. And that gives us a reference data for where the peaks are. And then when we're doing the statistics at finite temperature, we say, well, let's find which one is the closest to what this one was at 0 Kelvin, the one that is closest to this one, and so on. So that way we assign them to bins, and we're collecting statistics in the right bin. There's a reason why I'm telling you this story, so we're going to get back to this. But anyway, after we do that, we have constructed functions that are sensitive to the structure of the crystal we fitted it for. So now we can mix and match those functions and use them to construct other parameters. And we do it in different ways. So you can use these ones that are just based on distance. So here what you would be doing basically is building a model for the regular G of R, center of mass G of R. In practice, that's not usually enough. So we include orientational information. You can include relative orientations. You can include all three. There's different ways we can do it. And we typically try several, and we make a choice based on first how simple the function is, and second how good it is to separate the crystal from the solution or the melt. And we basically do it by constructing histograms of this function. And of course, one drawback of this is that you end up with a lot of functions. So in order to reduce the number of functions, we typically do a spatial decomposition of our simulation box, and then we take local averages in the subcells. So that way we give up a bit of resolution, but we also reduce the number of corrective variables. And here's one example. This was the very first calculation that I did. So this was for benzene, and what you're seeing there is a critical nucleus of benzene inside of a benzene melt. All right, so this is one example that is more realistic and more interesting than benzene. So we're looking at two sulfonamide drugs. These are sulfamerazine and sulfadiazine, and this was a project that was funded by Pfizer. These are old antibiotic drugs, so there's no IP involved, which means that we could publish what we did. And I find them interesting because, there are two molecules that are almost exactly the same molecule, but they behave relatively differently in the solid state. So this one is, has a solubility that's about a third of the other one. But also, sulfamerazine has 3 polymorphs, and sulfadiazine has 1, and that one doesn't look anything like the other, like the other 3. So that very tiny change of replacing a hydrogen with a methyl group here actually has a pretty big impact on the crystal structure. And, that's actually a common thing with crystals because, in the crystal, your molecules are packed very close together. So even small changes in the molecule can actually lead to a completely different structure becoming stable. So we were interested in this. And we did these simulations with the stream method for sulfamerazine. And we picked 2 forms. There is the other form 3 is fairly rare and not usually observed. So we picked the 2 that are more commonly observed. And we had a number of questions. We know that form 1 is a metastable one at room temperature. So the question is, when we do the nucleation at room temperature, does form 1 appear? That's what you would expect from the osmotic stages. or which one has the lowest nucleation barrier, which is what we would assume is the one that is most likely to crystallize. Here we did the fitting of the order parameters, and I'm just showing you, that this is how we test that the order parameters are able to differentiate between the structures. So we just construct histograms, and you can see that they are well separated. In this case, this is a log scale, so the typical ranges of values of something that is in a melt is very far from the crystal. So this is what the results often look like. So you have, this is a potential of mean force, which is we use as a proxy for the free energy. It's not the real free energy along the reaction coordinate, because we are actually doing this with a relatively large number of degrees of freedom. There's a way to correct for that, but in this case we didn't do it, mostly because we were interested in looking at many different solvents and different polymorphs, and it would take forever to do the whole story. But in this case, we use this potential of mean force as a measure of how easy it's going to crystallize this. And what you see basically is the molecules here are colored red if the local environment, as predicted by these functions I talked about before, looks like that of a liquid, and they are blue if it looks like that of a solid, right? So we did this this is sulfamerazine in methanol, and we did it for a number of solvents. So we did acetonitrile, methanol, and water. And we did the calculation for form 1 and for form 2. And this is the results that we obtained. So the two things to pay attention to here are that you always see a lower barrier for form 1 than for form 2. So what that is telling you is that most likely you would expect that if you do the crystallization at room temperature, you would observe form 1 forming, and not form 2, which is the thermodynamically stable one. And also you see that the barrier is lowest for methanol and largest for water, so that gives an indication that if you wanted to do this crystallization, you would want to do it in methanol because it will be easier to crystallize than in water. So we did, so the student who was working on this project was amazing, and he could, not only like me, was able to just do simulations, but he actually did experiments. So he went to Groton, to the research facility of Pfizer, and he did some experiments to try to validate his results. And one of the things he did was to actually carry out these crystallization experiments and do XRD. And he found that indeed, in all the three solvents, ambient conditions you get form 1, so that's consistent with the results. The other thing he did was nucleation induction times measurement. So they have this little machine that runs this experiment where you have basically multiple crystallizations happening simultaneously, and at every at various times you look at how many of them have crystallized, and you can use that to fit to a Poisson distribution and find what the nucleation rate is. So he actually did also these experiments and measured the nucleation rates, and what he found is that indeed you see the fastest rate when you do the crystallization in methanol, and the slowest when you do it in water. He actually had to add a little bit of acetonitrile, because in water the solubility is pretty low and the machine had trouble seeing the crystallites that form. So, but I mean, I am not going to make any attempts to say that I can quantitatively predict anything, because predicting nucleation rates is ridiculously hard. But what I am happy about here is that we are getting the right ordering, so we can actually at least qualitatively say, if I do it in these conditions, it's going to be easier than to do it in these conditions. Okay? So it indeed seems to be that the model captures enough of the physics to be able to tell us that information. So that's nice. Now the question is what we do next with this, and what we started asking ourselves is, what is the solvent doing? And the reason we're asking about the solvent is actually that the presence of a solute that is doing something in a solvent matrix actually can affect what the solvent is doing. And this is not a new idea. I mean, this is kind of, for example, the basis for the hydrophobic effect. This is an early calculation we did where we're looking at water surrounding a tripeptide, and there's a phenylalanine residue here, and the colors actually are an indication of how much the local structure of the solvent looks like the bulk solvent, and you can see that this phenylalanine has a massive effect on the water that is around it, and it actually goes, pretty far into the liquid. But the reason we started looking at this was, I was reading a paper by Matteo Salvalaglio, who's now at UCL. He was at that time working with Michele Parinello, and he was looking at nucleation of urea, which is Michele Parinello's favorite molecule for this. And he was looking at this in different solvents. And what he concluded from his simulations is that, if you do it in methanol and ethanol, the nucleation follows a classical mechanism. So what you have in this plot, I'm sorry, N naught is the number of molecules in the largest crystalline cluster in the system, whereas N is the number of molecules in the largest cluster in the system, without it necessarily being crystalline. So if you're moving along the diagonal in this plot, you're basically following a classical one-step nucleation mechanism. But if you are moving first up in the N direction and then going back to the diagonal, that is an indication that you're seeing a two-step mechanism. So what he concluded here is that basically just by changing the solvent, you're switching from one mechanism to the other. And I figured, well, that tells me that what I have been doing, which is only defining collective variables involving the solute, may not be enough to capture the whole picture of what is happening in this process. So I may be missing information that I need to construct the reaction coordinate if I'm only looking at the solute. So I figured, well, it would be neat then if I could do something similar to what I did, but for the solvent. And again, just like with the case of the solute, we want to be able to do this for multiple solvents, so we don't have to sit down and figure out how to define the parameter every time that somebody gives us a new solvent. So I wanted to adapt the same procedure that we were using before, but now for liquids. Sorry, I'm getting some weird notification. So so the idea is to again construct these functions again. But the problem we have now is, how do we collect the statistics? You remember I told you this story about, we took the structure at 0 Kelvin and then we compared to that structure, but for liquids that doesn't work because, you don't have a structure at 0 Kelvin. So this is not so trivial. So the way we ended up figuring that out is we take the entire molecular dynamics simulation, we collect all the data, and then we define a metric in this space of distance, boundary orientation, relative orientations, and we use it to do clustering in that space to find what is the best way to separate the data observed in the MD simulation into peaks. Okay, so that's what we do here. So we store in memory all the data of all the neighbor distributions that we observe at every time step that we collect statistics for, and then we figure out, based on that, how we assign it into peaks. And we play with the number of peaks. We did this with K-means, so we play with the number of peaks until we get something that gives us a reasonable description in terms of how compact the clusters are and how far they are from each other. Unfortunately, this is not as difficult to do in this case because this is a very high dimensional space, so the peaks tend up to, usually are very well separated. It's not so hard to do this. And we have a code to do this in GitHub, so if you want to play video games with it or whatever, it's right there. But this enables us now to construct these order parameters, but making them sensitive to the local structure of the solvent. So now if we put a solute there, we can ask ourselves, well, what is the solute doing to the solvent? And we can answer that question in a quantitative way. So Matteo was actually kind enough, and he shared the data from his metadynamic simulations with us. And this was the very early calculations we did. And we did these calculations where here the line representations are the solvent, and they're color based on these order parameters. And blue means that the local structure of that around that molecule looks like that of the bulk liquid, whereas red means that it doesn't. And what we observe here is that if you put an urea molecule in methanol, the methanol doesn't seem to care that much. It doesn't seem to have such a major effect on the local structure of the liquid. Whereas when you put an urea molecule in acetonitrile really doesn't like that. The local structure of the solvent there is disrupted, and that creates a driving force or aggregation that is not present in the case of methanol, and that leads to what we hypothesize that's what's leading to the two-step mechanism. So we've done this, and what I'm showing here is data that, you know, I told you that I have more questions than answers, and we have done a little better calculations here. But what we've been doing is doing histograms of these solvent order parameters at different concentrations of urea for the different solvents. And what we're hoping to see here is, you know, quantitatively, what is the solid doing. We want to calculate, for example, what is the entropy associated with these order parameters, and see, you know, if that entropy change is large or small, depending on which solvent we're looking at. So we've been doing these calculations. I actually have calculations for bigger systems than the ones I'm showing here, but they're relatively recent, so I haven't been able to make pretty pictures. However, when we've done these calculations, this is one of the early calculations we did, we run into a bit of a problem because not only you're seeing the effect of adding the solute, but in some cases you're also seeing some microphase separation happening in your simulation because you know, these molecules are fairly insoluble. So if your simulation box is not really big, you may end up with a supersaturation that is so high that you're crossing the spinodal and the system just kind of phase separates right away. So we're kind of right now trying to deconvolute those two effects. But, you know, like what these very early calculations were indicating is that you do see a larger change in the entropy associated with the solvent order parameters for acetonitrile and water than you do for the alcohols, which would be an indication that there is this entropic driving force for aggregation. But this is very preliminary, so as I said, you know, like hopefully you will be seeing something prettier from us about this. But I wanted to give you an idea of what direction we're going with this. The other thing we have, and I'm kind of at 25-minute line, so I'm going to finish very quickly, is some indirect evidence, and this is in the case of sulfadiazine, that if you incorporate solvent order parameters, those may also be important in describing the process. And you see it here where we have the gradient of the solvent order parameters along the nucleation pathway, and you see that there are jumps around the vicinity of the critical nucleus in some of the solvents, but not in the others. So that also tells you that there is something qualitatively different that is happening when you have different solvents. So I'm going to finish because I'm out of time. So this work was done in combination with a team from Pfizer, and I have a couple of students of NC State who are working now on the solvent stuff. And I always like to have a funny picture at the end of my presentation, so this is my funny picture. And again, thank you so much for the invitation and for listening to me. So thanks.
So thanks, Erik, for the wonderful and fantastic talk. So let's give Erik a round of applause. And so if you have any questions, just, you know, raise your hand, and basically you can then unmute and ask your question. So before people ask any questions, I was a bit fascinated by the comparison that you showed between nucleation barriers coming from simulations versus experimental rates. It looks like that the difference between experimental barriers, as you would infer from these rates, are much smaller, right? Like they're like probably less than 1 or 2 KT, right? So why do you think is that? Is it just luck that you get the order right, or is there anything deeper about the model that even though it exaggerates the difference, it gets the order correct?
So, well, I mean, like this goes back to the point I tried to make, that, you know, I don't make any claim that I can be quantitative about this, because, you know, with nucleation rates, even different experiments don't agree with each other, right? But yes. There's one thing we're doing here, and we're working at supersaturations that are much larger than the ones you see that you use in the experiments, right? And that's not because we want to, but because we have to, because otherwise you have to have so much solvent that you cannot run the simulation, right? So probably some part of the effect is the fact that we're working at much larger supersaturations, and they're probably not even the same supersaturation. Okay. So in that respect, you could be inclined to say that I'm lucky. You know, I always I always say that I don't complain when my results actually match the experiment, right? But there is, of course, a lot of other stuff that is happening here that we're not accounting for. And then there is also the issue that I kind of briefly touch when we're talking about solvents, that in these cases you run the risk of crossing the spinodal and then going into a regime where even what I'm trying to do, which is looking at a single nucleation event, doesn't make sense, because you may have a two-step mechanism, you may have nucleation happening everywhere at once. So there's a lot of simplifications we're making here. My point was just at least we're getting the ordering correctly, right? And we've done this for sulfadiazine, by the way, and we also get the ordering correctly in that case. So at least for these two, we're getting something. We're getting enough of this right that it's working. Yeah.
That's fantastic.
Thank you.
So I think Sapna has a question. Sapna, you can unmute and.
Sure. Thanks, Erik, for that wonderful talk. Since you're on slide 19, if you go to slide 20, I just had a clarification question.
Mm-hmm.
What is the different color coding here for the solvent?
Yeah, so this was one of the early tests we did with the solvent order parameters. So these are waters, and they're colored red if the local environment is very different from bulk water, and they're blue if it is very similar. So if you see red, that means that there's something there that is making water unhappy, or it's being forced into an entropically unfavorable conformation.
Okay. And then I had a question on your slide 29. Where you showed the solvent effects, and you showed that some, the gradient of your solvent order parameter is related to the nucleation barrier. In some cases, in some it isn't. But do you know what that implies physically? As in, do you know how the solvent is affecting that, or what's going on?
Yeah, so what this is telling me is that, so if you think about what this gradient is it's telling you how much the structure of the solvent is changing at that point, right? So, in one case, you see something that is just monotonic, right? This is what you see in methanol. In the case of acetonitrile, you see that it kind of goes up, goes down, and then goes back up. So it's kind of, it's kind of hard to be specific about exactly what is happening, but what this is telling me is that there is some substantial rearrangement of the solvent in the vicinity of the nucleation event. That is happening in acetonitrile, but it's not so much happening in methanol. Okay, so this nucleation event that is happening is really causing the solvent to have to do something, and that is why I wonder whether we really need to include explicitly these order parameters in our description, because at the end of the day, what we're going to end up hopefully getting is some approximation of the reaction coordinate as a function of the other parameters. But if I'm looking at a subspace that doesn't contain all the information, then maybe it's not right. Right. So yeah.
Okay, cool. Thank you.
Mm-hmm. So I think I don't see any other questions. So I think we are almost out of time. So I would let's thank Erik again.
Thank you.
And we can, very fantastic work. We can move on to Kellen. So Kellen, you can take it up from here and introduce the second speaker.